I can see Jeff here. Good morning, Jeff. Can you hear me?
I can hear you. Can you hear me okay?
Yes, perfect. So without any further delay, I would like to introduce Professor Jeff Lichtman, the Jeremy Knowles Professor of Molecular and Cellular Biology, and the Ramon Cajal Professor of Faculty of Arts and Science at Harvard University, who I had the privilege and honor to work with during my sabbatical at Harvard. My admiration for Jeff and his immense contribution to the neuroscience field finds parallel with the Spanish neuroscientist from the 19th century, Don Santiago Ramon Cajal, who, with very rudimentary microscopes, hard work, and countless hours of careful observations, made the first and most accurate description of how neurons physically communicate with each other. Jeff, certainly inspired by Cajal's works, developed new techniques such as the brain-bone transgenic mice to visualize neural connections and monitor how they are altered over time. Recently, his efforts have focused on developing new 3D electron microscopy techniques to map neural connections of the developing and adult brain. Jeff's key contributions can be found in his many articles published in the most respected scientific journals like Nature, Cell, Neuron, and others. More than a century separate these two great minds of the neuroscience field, and still the underpinning of the cellular structure of the brain is not well understood. However, as we will learn shortly, with Jeff's contributions, especially concern electron microscopy methods, we may be better poised to address these big mysteries of the brain science. Jeff, thank you so much for accepting our invitation and for taking time for giving your talk.
Thank you very much. Thank you very much for that kind introduction, Christina. It's a pleasure to talk to you.
Likewise.
Can you see, is my screen on? Connectome perspective at the top? Good.
Yes. Mute me.
I've been an imager, a biological imager, since I was a graduate student. And although I've done some physiology, I think most of the insights that I regard as believable or strongly believable come from what has come in through my eyeballs. And connectomics is a field I'll talk some about. That perspective is very strongly related to imaging. And as you've already heard, neurobiology in general and connectomics in particular begins with Ramon Cajal. And I want to just say a few words about him because it will stimulate a particular mind view, the perspective I'm trying to get across here. Cajal, as you've heard, is a really remarkable genius, certainly the greatest neuroanatomist who ever lived. Here's a picture of him in his laboratory with 3 microscopes. I think if you can you see my pointer? My pointer show up there? Yes, you can. A little glass of sherry right here, which nowadays would be considered absolutely not allowed, given especially the wide range of toxic chemicals he used. He had one of the first apochromatic lenses that Zeiss made, and this is one of the reasons he could see better than his colleague and competitor, Golgi, who developed a stain that Cajal used to elucidate a lot of deep mysteries about the brain that Golgi denied really were visible there. And in fact, they even had an argument at the Nobel ceremony that they shared. The Nobel Prize they shared. He used Cajal's Golgi stain to study the nervous system and elucidate many fundamental principles of the nervous system's organization. He was a visual genius in the sense that he could see something and then remember it and then draw it back. And his drawings are just fantastic. They're masterpieces. This is a piece of cerebral cortex stained with the Golgi stain. And the most notable thing about the Golgi stain is its inefficiency. Even though the brain is filled with neurons, the Golgi stain stains maybe 1% of them. for interesting reasons. It's just a very unlikely reaction that occurs every once in a while inside a cell. And the crystals that form of silver dichromate can't pass through membranes. So once they start to crystallize, they fill up the cell's many processes and give you these beautiful kinds of stains. If the Golgi stain was more efficient and every cell stained, it would have been useless. You wouldn't have been able to see the forest for the trees because the trees would all be labeled and therefore you wouldn't be able to see them as individuals. But because he could see them as individuals, he could not only take drawings of them, but he could begin to learn details about them. And here's a classic Cajal picture. And the thing to notice here, in addition to the fact that the paper got yellow over time, but he used a kind of whiteout to make corrections, very, very subtle corrections that we would never notice except by virtue of the fact that the whiteout lasted longer than the paper. But that's not the big thing about this picture, this Golgi stain picture of cerebral cortex. It was these little arrows, which was Cajal's idea of how information would flow through the nervous system. So if we start down here, this process is coming out of a cell from another part of the brain. It's an axon, which leaves a cell and sends information to other cells we now know. And according to Cajal, it terminates on top of these fatter branches known as dendrites of a neuron. And according to Cajal, information passes from the axon into the dendrites. That information flows into the cell body of the target cell, if you will, and then the target cell sends that information out its own axon. Notice these arrows up here, which then overlap with the dendrites of another neuron, this neuron A. And at the same time, here is another axon from another part of the brain contacting the basal dendrites, as they're called, of this very special type of cell in the cortex known as a pyramidal cell, whose cell body is sort of shaped like a pyramid, a little bit triangular. And together, Cajal said, these two inputs collaborate in some way to generate a signal that leaves this cell out its axon. Notice these arrows here. And that axon touches the apical dendrite of this pyramidal cell, and that information flows into the cell body and out its axon, and the axon does the same thing over here. So here in one fell swoop, in one very short picture, he's explaining how information might flow through neurons in the cortex. And this had a profound influence on the way people have thought about the nervous system ever since. In fact, this sparse worldview, where you just see a few cells, have led people to be able to draw sort of cartoon drawings of how the nervous system works. This sparse worldview based on the Golgi stain and similar techniques are the basis of most of the cartoons of the types of cells that are interconnected rather than diagrams of actual networks. So here, for example, is a picture from Rudolfo Yinas and colleagues of the way cells in the lowest layer of the cortex, layer 6, these pyramidal neurons, which send their axon back down into the thalamus, send information, notice these arrows here, which contact the dendrites of cells in the thalamus that send their axons back up to the apical dendrites of pyramidal cells, and added something that Cajal didn't know about is inhibitory neurons, neurons that in fact try to stop information from flowing. So these negative signs as well as positive signs. But with that minor difference, there's a whole world of neuroscience based on building diagrams like this of the way the nervous system works. There is only one problem with this, is that this is not a neural network. This is a cartoon based on sparse labeling. And so it remains an open question, what do networks actually look like? What do they really look like? And that is one of the main reasons it's useful to think about imaging techniques that allow us to see the connections, not just of a sparse subset of cells, but exactly how many cells are sending information to a target cell and how many target cells are being contacted by the target cell itself. So what is the fan in and fan out of each part of a neural network? And this is a general question about why people do synapse level connectomics. And there are two general sort of points of view here as well. I just want to mention them briefly because the rationale of why you do something sometimes explains what you do with a technique. And there's one view which is very reductionist about why one might want to do synapse level connectomics. And that's, instead of thinking about the forest, you think about the trees, the individual entities in the nervous system. So this tree view, you might want to get a detailed connectivity map of various nervous systems or parts of a nervous systems as a means of getting several kinds of information. One is you would have exactly the wiring diagram that underlied a particular behavior. If you imagine you're driving a car and you see a red light and you automatically, without thinking, put your foot on the brake pedal, that's presumably because information is flowing from the red sensitive photoreceptors all the way to the muscles in your leg that are moving your leg from the gas pedal to the brake pedal. That's a connectomic question, but how that happens would require tracing all those wires. So one reason of doing connectivity maps is to get exactly what the wiring diagram is for very complicated behaviors. Another reason is that there are a lot of diseases probably where the wiring is not normal. We suspect this, actually. Humans suspect that there are a number of diseases where wiring is abnormal, perhaps some of the autism spectrum disorders, or perhaps diseases like schizophrenia or Tourette's syndrome, or might be diseases that could be called connectional pathologies or connectopathies. And one might get insight into them if one has animal models and see a wiring diagram that is discretely different from a wild type one. And a third is there's a huge interest now in artificial intelligence. But if you'd like to understand intelligence in its biological form, you're going to have to look at the machine that allows for that kind of intelligence. So if you want to simulate biological intelligence in silico, you might want to imitate exactly what the wiring diagram of an intelligent animal is. So those are all very fine structured results that might come out of connectivity, but there's also a broader view of what else one might do or what might one do instead of these very detailed maps. And one is you might derive principles of neural circuit organization as opposed to specific neural circuits. These principles would be a means of understanding, for example, how experience, which modifies and in some cases molds behaviors, as in human beings, where you learn to take your foot off the gas and put it on the brake when you see a red light. That wasn't built into your nervous system. You somehow learned to do that. If you want to understand experience-based circuits, you're going to have to learn the principles because every animal and every person's experiences are different. So you're not going to find a canonical circuit for doing that because each person is learning specific things based on their own experiences, not only people, but animals as well. So that's one sort of principle view, forest view. Another is to understand how circuits form in the first place. How do you get this extraordinarily complicated brain out of a very modest beginning, a fertilized egg cell? How does that happen? And you could do connectomics across development. And finally, if you wanted to build intelligent circuits, you might learn a lot by looking at the principles that one built intelligence in us. Not so much to simulate exactly what an animal does, but to understand the principles well enough that we can build intelligent circuits based on those principles. My own heart is closer to the forest view than the tree view. The big questions are what I'm most interested in. And some of them include in what physical form are the behaviors that we learn encoded in our brain, and how do they get there? And that basically is, how does the experience of doing something change the brain's structure? So how does the brain's function, which is how it functions during experience, create a structure that holds a permanent map, a memory, if you will? How is memory stored? In what form does it take? a shape in a brain. Nobody knows the answers to questions like this. And given that the brain is mainly a vast set of nerve cells connected by trillions of synapses, these questions are one place where the new field of connectomics may be useful. And by connectomics here, I'm basically saying mapping out all the wires that connect nerve cells. I want to tell you two short stories about this relationship between experience and wiring diagrams. And I'm emphasizing imaging, how imaging has answered these questions for us. And the first one, the first story that I'd like to talk about is a longitudinal study of connectomics in the neuromuscular system of mice. So this is developmental connectomics. And this is work of a number of people. Yaron Mirovich and K. Kang and Ryan Draft are the people who did the hardest part of this work. But there is a whole host of other people who are working on this as well. This work is in bioRxiv at present, and we're getting ready to submit it. But I'd like to talk about this work, and I'm going to start not talking about 3D serial electron microscopy, but light microscopy, confocal microscopy. We go back and forth between these techniques because sometimes serial electron microscopy is just too difficult because the data is too large. So let me show you a very small adult muscles connectome. That is all the wires that connect nerve cells to muscle fibers in a small muscle that pulls the mouse ear back. It's called the interscutularis muscle. And this is a tracing, basically, of the adult interscutularis muscle's connectome. Each of the axons going to this muscle come from a different motor neuron. So each color is the branches of a different nerve cell in the spinal cord. And this was made by serial reconstruction from confocal microscopes. In many cases, we use brainbow animals to do this. I'll talk a little bit more about brainbow in a moment. In this case, it was large enough that we could use a monochromatic fluorescent label, and then the colors were superimposed on afterwards. The things are big enough that we get no confusion. And you see this big nerve at the bottom here sending branches that go to a muscle. The muscle is about 1 millimeter from here to here. And these sort of pretzel shape affairs are the neuromuscular junctions. So that's where the nerves release neurotransmitter to cause muscle fibers to contract. The muscle fibers are running roughly in the direction that I'm pointing back and forth like this. They're not shown in this picture. So if we look at one of these neuromuscular junctions at higher power, you see the axon comes in and it branches on the muscle, and there's a little red outline here. That's where there are acetylcholine receptors, the receptors on the muscle fiber that respond to the chemical being released by the nerve, the chemical being acetylcholine. And so each neuromuscular junction is really unique in its shape. They each one have its own particular shape. Every one is different. We call these sometimes end plates, and they're sometimes called NMJ for neuromuscular junction. All those terms mean the same thing. And there's a single axon coming in. So it's a neuromuscular junction or end plate, and it's singly innervated. Every one of these is singly innervated. That means there's only one axon going to that muscle at that site. And in fact, most muscle fibers only have one such site. Most muscle fibers have just one neuromuscular junction on their surface. So even though the fiber extends a very long distance in both directions, from here all the way up to here, most muscle fibers, but as you'll hear me say, not all, but most muscle fibers only have one neuromuscular junction. And that junction is powerful enough that when the nerve sends an electrical signal into the terminal, that's enough to make the muscle fiber contract. So the axon that is releasing neurotransmitter has a powerful, what's called suprathreshold quantal content. It releases enough vesicles of neurotransmitter to invariably cause the muscle fiber to contract. So this branch is only one branch of that light green axon. If you look down here, here's another branch of the same axon. And up here, there's probably another branch as well. If you take all the branches together, That's what that motor neuron does to the muscle. It causes a number of muscle fibers to contract. And that's called a motor unit. And I'm sorry for all this biology, neurobiology terms, but I have to use them. It won't make sense otherwise what I'm going to say. A motor unit is the set of muscle fibers innervated by a motor axon. By the way, if anybody is confused and I'm saying something that's very confusing, don't hesitate to interrupt me. I'm happy to take a question or two if you have questions. So a motor unit is the set of muscle fibers innervated by a motor axon. And motor units in a muscle range in size from some that are very large, that mean that they contact lots of muscle fibers, to some that are very small that contact a few. And there's a arrangement to these, not in terms of position in the muscle, but in terms of function. The large ones, which cause large contractions because they contact many muscle fibers, are usually recruited after the small ones. So the small ones cause small muscle contractions and maintain muscle tone, and the large ones are used when the muscle has to use maximum strength. Motor units are thought to be recruited in a fixed order from the smallest motor units first to the largest one to give a graded contraction. This is sometimes called the size principle of Henneman. Here's a little diagram of this. I'm going to show you now. This is not an actual biological result, but you would get this result if you did this. If you ask someone to exert more and more force in a muscle, they will start by recruiting the weakest motor neuron, which causes the smallest contraction. And if that's not sufficient, another axon will be recruited on top of it. So here we have a bunch of motor units, starting with a small one and ending with a large one. And I'm going to show you what happens over time as the tension and therefore the constriction of the muscle increases. So first, the smallest one fires, then the next one fires, then the next one fires, and then the next one fires. And if you look up here, the smallest one generates a certain amount of tension in the muscle. When the second one is recruited, the tension increases, and it increases by a greater amount than this original step because this bigger motor unit contacts more muscle fibers. Then the third one contracts, and that adds even more tension, and then the fourth one, more tension, and so on. There might be 20 motor units or so, meaning that you have very fine control of fine movements, but by the time you get to maximum tension, the muscle is much more crude, and that is generally the way we know. We have much more finesse when we're doing fine movements than when we're using maximum tension. So let's look at these motor units one at a time in a muscle. Here's one motor unit, and here's another motor unit. And you see that the axons, which end in neuromuscular junctions, are interdigitated in some sort of random pattern. I hope this is playing okay, that it comes in and you see them come in one by one.
It is.
Okay, good. And these are kind of small motor units and are getting smaller. We started with the biggest one. I'm putting them in reverse order here, and we're going to end up with the smallest motor unit. And you see that there's nothing you can really say. It seems sort of random. And indeed, if you do the same test which Julu did in this paper from PLOS Biology, every specific muscle has its own wiring diagram. Even the same muscle, if we did 12 of these, the left and the right motor unit, the right muscle, and we did that over and over again for animal after animal, which we did for 12 animals, everyone had a slightly different wiring diagram. So that is the, if you will, the connectional map or the connectome of this little muscle. To start to think about this in a more quantitative way, we have to graph this. So we have to somehow graph this wiring diagram in a way that we can begin to do analysis. And the graphing approach we've come up with is pretty straightforward. We just make a circle that has all the muscle fibers as little dots. The size of the dot is related to the diameter of the muscle fibers, but they're all so similar you hardly can see the difference in the graph I'm going to show you. And outside of that circle, we have a circle of the motor neurons and allowing them to connect to which muscle fibers they wish to connect to. So this is the same wiring diagram, but now in graphical form, where the inner little dots are the muscle fibers and the outer dots are the axons. Let me just go back here. The outer dots are the axons, and this is the biggest motor unit. And so it has branches to more muscle fibers than any other. And the smallest motor unit is here, And there's about a 10-fold difference in the size of the biggest to the smallest in the adult. So that's the wiring diagram of an adult. And most of these muscle fibers only have one line going to them because most muscle fibers only have one axon innervating them. As I said, I'll get back to that idea again a little bit later. Now let's just do the connectome at birth. This had never been done before, and it was hard to do because the size was so small. We couldn't use light microscopy. So we used serial electron microscopy. So I'm going to show you the same kind of diagram I just showed you for the adult, but now for the baby. And the first thing I'd like you to notice is sort of the pattern of the branching here, and compare that to the pattern of the branching in the baby. So that is the smallest motor unit. And then that's the next smallest. And as the colors get redder, that we'll be getting to larger motor units. And the axon is coming, the axon bundle is coming in from here. We're adding more and more. And I think to a first approximation, the branching pattern is not fundamentally different from the adult branching pattern. That is, you see the same kind of structure to the big branching. It may look a little hairier, and I'll talk about that because that is the main difference. That is, there may be more side branches, but the total architecture of the branching pattern to the muscle is no different in babies than adults. So the adult branching pattern is basically generated by the baby branching pattern. This is at birth, and nothing much happens to the general branching pattern. So now we're getting to the larger motor units. And they are beginning to look a lot like the whole branching pattern. It's like this each single new axon is just sort of fitting into the entire pattern and largely replicating it, a little different from in the adult where they look like trees. Here, the individual branching pattern more reflect the entire branching pattern. And let me just put this in the context of the muscle for you. So here's the muscle. You can see down here that one of the branches dives very deep, so it's hard to see. But generally, this is a branching pattern in this little interscutularis muscle that's not very different from the adult, but there is a profound difference. And if we go to a single neuromuscular junction, you can already tell, looking at that little green box, that there's a lot of colors there, not just one color. And we do serial EM, so we can look at a serial EM picture of a single neuromuscular junction. This is work that Juan Carlos Tapia did of a single muscle fiber with serial electron microscopy, where we take a section after a section after a section through a muscle. And then he colored in the muscle green and colored in each of the axons going to this one muscle a different color. And then you can render from that tracing all the axons. And unlike the adult, where there's a single axon going to a neuromuscular junction, now you have a neuromuscular junction with a whole bunch of axons going to it. And here they are. That's the axon that has the most territory. And I'm putting them in one by one from the biggest axon to the smallest. Now, we're not talking about the branching of a motor unit. We're just talking about the branches of axons to a single muscle fiber. And that last little pink one is actually already retracting. So if we do the wiring diagram of the whole muscle now, which clearly has more than one axon going to each neuromuscular junction, the wiring diagram looks different. So this is the adult wiring diagram I've already showed you. Now let's look at the baby wiring diagram at birth. And, you know, this is a baby mouse, so I suspect the same would be true for a baby human being. There's no question this is a profoundly different wiring diagram in babies than in adults. You have the same number of motor neurons, same number of muscle fibers, but now the number of branches these axons make is profoundly larger, although still there's about a 10-fold difference between the biggest one and the smallest one, but now every axon is innervating lots of muscle fibers. So I hope that's clear. Is that reasonably clear? I can't actually tell because I can't see anybody's face, but if someone needs to ask a question now, this would be a good time to ask a question about this. Okay, so all neuromuscular junctions in babies are multiply innervated. They have more than one axon innervating them, and maybe against your intuition, when you go from being a baby to adult, your nervous system is getting simpler, not more complicated. And that is an important point here. So one conclusion one can make is that given the randomness of where the branches end up, you can say that we have one connectome at different ages. In the adult, we have strong, very strong, powerful, intentional input of one axon that got lots of release sites on one muscle fiber. And that emerges from very weak connections that are sort of random and almost everywhere. About half of every possible connection that could exist exists in a baby. So you have this weak sort of incidental inputs in development, and somehow you go from the weak many connections to the one strong one on every single muscle fiber. And I can't help but wonder if learning, early experience being the learning in this case, transforms these many weak connections to a few strong ones. That is the question I'm sort of after here. And it's useful then to go from birth to an intermediate stage where some of the connections have already been eliminated, to see if we can get any insights into this process by imaging an intermediate time point between the adult connectome that I've already shown you and the connectome where almost everything is connected to everything at birth. So how does the neuromuscular junction transition from many weak to one strong input? And for this, we used a technique that we developed a few years earlier called Brainbow, where we use, it's basically a Technicolor Golgi. It allows us to do the same thing Golgi stain does, image individual cells in their entirety, but because each cell is a different color, we can much more densely label our tissue than was possible with Golgi, where if everything was labeled brown, you wouldn't be able to distinguish one cell from another. Brainbow is a transgenic approach that gives a random subset of the amount of red, green, and blue fluorescent protein, for example, in each nerve cell. So each nerve cell has its own RGB hue. So we began doing Brainbow imaging at 1 week of age, and this is a confocal stack. Many of you are probably familiar with confocal microscopy. It's a wonderful technique because as you focus up and down through a tissue, out of focus material gets filtered out by the pinhole in the return light path. So you don't see blur, you just see sharp images of each focal plane. And all of these are, again, it's a wiring diagram. So all of these are neuromuscular junctions. And I think if you look carefully, you'll notice many of these neuromuscular junctions are multicolor. They have more than one color. That one up there where I was pointing was green and purple. If you look around, you'll see there are a lot of multiply innervated ones, but it doesn't look like there are 10 axons. Many few are here. So confocal optical sections disambiguate connectivity at 1 week. At this age, about half the neuromuscular junctions are already singly innervated, and the rest are contacted by 2 or more different axons, but many less than 10 or so that we saw at birth. But pruning is not yet complete, and we hope this would give us some insight. So let me just zoom in on one little region here. At 1 week of age, there are fewer multiple axons sharing each neuromuscular junction on developing muscle fibers, and often the inputs that remain are unequal in size. So for example, this neuromuscular junction here is orange, but there is a grayish axon that has a retraction bulb here. And this neuromuscular junction is mostly contacted by the red axon, but there is this grayish axon as a small territory. Here's a neuromuscular junction where it's about 50-50, and here's one that's maybe 60-40% red and green. And we see all, basically can get every single neuromuscular junction in a muscle with connectomics. This is work that Ryan Draft did that's part of this larger study that I've been telling you about when he was a graduate student. And one of the things that was clear after looking at a whole muscle is that the connections that are maintained, that's the ones that have not yet been eliminated, are not a random subset of what was there earlier. I'll give you one example here from one of his mice. There's a little muscle in the neck, not the ear muscle, but a similar muscle with a lot of multiply innervated neuromuscular junctions with brain bow. And what's shown here is a bunch of muscle fibers that have the green axon and the purple axon at the same neuromuscular junction, many neuromuscular junctions like that. Although the green also shared muscle fibers with orange and with purple, these were easy to find. So the green axon innervates 29 junctions altogether. 7 of them are singly innervated, just where all the other inputs have been eliminated, but 15 junctions are still shared with other axons. 15 of the remaining 22 junctions that share branches, 68% of the remaining ones share the neuromuscular junction with one particular axon, the purple axon. And the chance of that happening by chance is, given the number of branches each of those axons have, very, very small. So here is the actual data. This green axon shares 15 muscle fibers, neuromuscular junctions with the purple axon. It shares 4 neuromuscular junctions with the orange axon, 3 neuromuscular junctions with the blue axon, and no neuromuscular junctions with the pink axon, which has many branches in this muscle, none with the violet axon, which also has many branches in the muscle, none with the khaki axon, none with the red axon, and none with the mauve axon. The probability of this kind of distribution happening by random chance is 0, basically, infinitesimally small. So the connectivity that remains is not a random etching away of axons from neuromuscular junctions. It's a highly non-random connectivity. And so that is maybe where you'd leave it, but because you have connectomic data, you can analyze every connection of every axon. And that's what connectomics does, is it provides the opportunity to see everything and see if there are some principles that emerge. So let me show you this for one particular muscle. I'm going to go again to a slightly different graphical approach to look at this. So this is a graph of all the multiply innervated muscle fibers that are innervated by an axon, the axon that is red, and how many muscle fibers the red axon shares with the yellow axon, which is 66. And this same axon, just similar to the case I showed you, shares fewer junctions with the green axon, in this case 29, fewer still with the blue axon. It shares 16 neuromuscular junctions, 4 with the blue, the darker blue, and only 1 with this purple axon. So this is a kind of gradient that it seems to have a harder time winning against yellow than it does against purple. You know, one or the other is going to win here, but there's very little multiple innervation left where red and purple share, even though we know in earlier life, everything was connected together. So I'm just going to show you this now in a slightly different form, which is the same exact data. It's just the actual data here, where each horizontal line is a set of axons innervating a single neuromuscular junction. So if a neuromuscular junction is innervated by the red axon and the yellow axon, it's this horizontal line here at the top that's red and then turns yellow. And red has lots of muscle fibers it shares with yellow. As I mentioned, it shares fewer muscle fibers with the green and fewer still with the light blue, fewer still with the dark blue, and only 1 muscle fiber with the purple. There are a couple of muscle fibers that are triply innervated, but interestingly, again, it's not a random set of triples. This is red, yellow, and green. In this particular order, this is the gradient of preferences, you might say, the red axon has for maintaining innervation with the yellow, the green, the blue, the dark blue, and the purple. Now we can do the same exact map for all the muscle fibers that have a yellow axon and they're multiply innervated, which would include the red, but also include muscle fibers that are not innervated by the red axon. So let me show you that pattern, and I'm not going to change the order up here. I'm going to leave the order the same. And you see basically the same kind of result. Yellow has a lot of preference of remaining multiply innervated with red. And also in the same order, which I haven't changed, it prefers green to light blue, and it prefers light blue to dark blue, and yellow has no connections with purple at all. So again, there's this interesting gradient, and there, again, here are some of those triples that I have already mentioned. We do the same for green. We have the same exact situation. I haven't changed the order up here, but green, we've already noticed, has a lot of affinity for yellow, but it also maintains a lot of connections with light blue, fewer with dark blue, fewer with purple, and relatively fewer also with red. If we go to light blue, the gradient is still the same. The order is still the same. Its preferences are blue, dark blue, and green more than yellow, purple, red, or purple. And if we do dark blue, it's the same story again. Its preferences are for its immediate neighbors in this particular rank order. Whatever this rank order is, it works for the wiring diagram of every axon in this muscle. And if we go to purple, it's the same as again. The purple prefers to multiply innervate muscle fibers at this intermediate stage with blue, less so with light blue, less so with green, not at all with yellow, and one with red. So that's a big mystery. And it wasn't just this one muscle. When we looked later in the developmental process, we found the same thing, but there was less sharing. Most of the muscle fibers were now singly innervated, but the sharing was, again, in this rank order. And when we looked at earlier stages using the same rainbow technique, we saw the same thing, but it was messier. Again, the preferences were mainly for your immediate neighbors, but there was also preferences that extended farther. There was more multiple innervation. So what does all this ranking mean? Unfortunately, it would take me an hour or so to give you the full story, but I can tell you we discovered what it meant in the most weird way of all, and it was imaging not P6, P7, P8 neuromuscular junctions or baby muscle, but looking at the adult. We got a hint from what at first seemed like a mild inconvenience in the adult connectome. Just by looking, we saw that occasionally muscle fibers in the adult have more than one neuromuscular junction. They should only have one, that's what the textbooks say, but we found about 6% of the muscle fibers had two neuromuscular junctions, and we learned that the timing of activity determined which synapse was eliminated in a syn-fire chain, which was the recruitment order. That last sentence will mean nothing to anybody. I'm just going to try to explain what I mean by how activity generated the maintenance of two neuromuscular junctions on some adult muscle fibers. So these adult muscle fibers look like this. They're often muscle fibers where the neuromuscular junctions are very far apart from each other, but there are clearly two on the same muscle fiber. Again, connectomics is necessary to do this because you've got to trace every axon and every muscle fiber. And when we looked at for the muscle fibers that had more than one neuromuscular junction, if the first neuromuscular junction was put here and they were all aligned here, the other one were reasonably far apart from where the first one is. The two neuromuscular junctions are always far away from each other. Interestingly, we discovered, not expecting to discover this at all, just by observation, a new form of synapse elimination, which is the loss of whole neuromuscular junctions with age. So if you make a graph like this for each age, you discover that at birth, there are a lot, 40% of the muscle fibers in this muscle have two neuromuscular junctions. And in the adult, it's down between 5 and 10%. And interestingly and importantly, whereas in the adult, the two neuromuscular junctions are very far apart, in babies, they can be very close together. And it is the nearby neuromuscular junctions that get eliminated and a subset of the ones that are far apart that are maintained. So there is some aspect of distance that is preventing two neuromuscular junctions from being on the same muscle fiber at the same time. I'm going to cut a very long story short here so I can go to the other story I wanted to talk about by just saying what we think is going on here. And it is quite surprising. It's that if you have two neuromuscular junctions on the same muscle fiber and they're not active at the same time, or they're very close together, so the time of transit of a signal from one junction to the other is short, you'll have situations like this where one action potential will be fired in an axon. It'll generate an action potential in the muscle, which will propagate to the other site. And that will happen multiple times. And eventually that will lead to the, by competition, basically, to the disappearance of the other neuromuscular junction. Activity that is occurring when a postsynaptic site is silent leads to its elimination. That's something we already knew. However, if you have two axons that have similar activity, their action potentials will collide, and action potentials can't pass each other. It's just a neuroscience fact that there's a refractory period after an action potential. So if you have two action potentials moving towards each other along a muscle fiber or an axon, they'll collide. So that means that one neuromuscular junction cannot affect the other neuromuscular junction, and they can remain. So that might give you the impression that axons that fire together wire together. It's sort of Hebb's postulate, but for a very weird reason. Hebb's postulate is that neurons that get associations from two inputs at the same time maintain those associations here. In this case, because of collision, they maintain their connection. So this is interesting, but why is it relevant to synapse elimination at a neuromuscular junction? And it's because of that size principle I mentioned before. Axons are active in a particular order. Axon 1 will fire before axon 2 and axon 2 before axon 3. So if two axons have a similar activity pattern and they're on the same muscle fiber, then their activity will cause a collision and they will maintain those connections. And axon 2 might, for example, be innervated at two sites by the same axon. Again, there'd be a collision and they'll be maintained. And axon 2 and 3 might have the same firing pattern, and therefore they would be maintained because of a collision. However, axon 1, axon activity pattern similar enough for two neuromuscular junctions to persist on the same muscle fiber owing to potential collisions might allow you to have a chain of connectivity like this in the adult. Whereas axon 1 and 3's activity pattern, because they're recruited in order, might be different enough that axon 1 fires long enough before axon 3 that will lead ultimately to the elimination of the other input. So that was the theory, and the facts bore that out remarkably. Putting this all together by looking at the connectome of the multiple end-plated adult muscle fibers, we found exactly this. This is really the data where there really is a chain of firing where axons are behaving as if their firing pattern is related to the timing of activity. Now, activity patterns similar enough for two neuromuscular junctions to persist on the same muscle fiber owing to collisions, what does that have to do with anything? This syn-fire chain is sort of a synchronous chain of activity that always forms in the same order. This is exactly the same kind of ranking that we saw at the multiply innervated neuromuscular junctions at 1 week of age, the Brainbow result I already told you about, suggesting recruitment order of firing is driving synapse elimination, both by eliminating neuromuscular junctions, but also eliminating axons whose firing pattern is different from the firing pattern of the axons that are maintained. And this is, for us, a deep insight into synapse elimination. It's not the amount of activity. It's when you fire relative to other axons that determine whether you stay or go. And of course, that is interesting because it also may be why synaptic rearrangements in the brain work the same way. So it raises the question, this is muscle, and I think most people say, well, if you're interested in learning, why are you looking at something as stupid as a muscle? And it raises the question, is simple connectivity in adult neuromuscular system, can that be useful when thinking about the insane complexity of the adult CNS. In muscle, we have one or rarely two axons innervating the same postsynaptic cell. There's only one type of postsynaptic cell, basically several classes of muscle fiber types, but they're all muscle fibers, and there's only a few types of axons. There's no inhibition. Everything uses the same neurotransmitter. Can we learn anything about the brain where you have thousands of inputs and outputs for each neuron, hundreds of types of pre and postsynaptic cells? You have inhibition, electrical synapses, modulators, many neurotransmitter types. So it might be hopeless. Nonetheless, we looked. And let me explain where we looked. We looked in an adult data set of the adult brain of a human being. This is a 1.4 petabyte, that's 1,400 terabytes of data that came from a woman who has epilepsy that was treated by neurosurgery. And this is a piece of overlying cerebral cortex that was removed from her brain to remove deeper in the hippocampus the epileptic focus. So this is normal tissue, histopathologically speaking. And the tissue you see there has color in it because we worked on this tissue with colleagues at Google who segmented all the data. So we had a serial electron microscopy data set of images that came off of a multi-beam scanning electron microscope. And then they segmented using machine learning all of the processes in there. So let me just zoom up on a part of this, give you a sense of what this data looks like. These circles here are cells. Some of them are neurons, some are glia. And this colored stuff in between is the neuropil, where nerve cells connect to each other. And the things that are not labeled are blood vessels, where my pointer is here. I'll just zoom up on this a little bit better, a little bit more, so you get a better sense of what this data looks like. You begin to see that these cells have branches coming out of them. Many of them are neurons. Some of these are apical branches of pyramidal neurons, such as this one and this one and this one. Zoom up a little more. You see sometimes a neuron is right next to a couple of other littler cells. This is a neuron cell body. Here's a few other cells. Cells like this that have lots of color in the same section are almost certainly astrocytes that are glial cells that make lots of little branches. So all this yellow you, I hope, can see in this image are branches of this astrocyte. These things, again, that are not labeled are blood vessels. Zoom up a little more. You can begin, I hope, when this finally refreshes for you, and I don't know when you'll actually see this image. It takes a second, I know, to load. These dark outlined objects are myelinated axons, all these little dark circles. Zoom up a little more. You begin to see the nucleus of cells, you see lots of myelinated branches, and you might begin to catch some of these double colored lines here, which are machine learning identified synapses in the data set. I'll zoom up a little more, and it's a little more clear that this is a synapse. That's a synapse. That's a synapse. That's a synapse. Zoom up a little more. So the data looks beautiful. I'll zoom up a little bit more than that, in the sense that every process of every cell is labeled a different color, not with Brainbow now, but with post artificial intelligence basically tracing these wires out. This data set is online, and you can do exactly what I'm doing, and you can click on any of these objects and generate the three-dimensional version of each and every object in here, which is quite fun to do. It's at Google. If you look under petascale map of human brain or something, you'll be able to find it. So if you render all 52,000 cells in the volume, it's a pretty messy looking piece of tissue. But if you color code these cells by size, simply by the size of the cell body, where the largest cells are red and the smallest are blue, you see that this piece of cerebral cortex is highly organized into layers, where layer 1, which is closest to the skull, has very few neurons in it. Mostly these are supporting cells called glia. This is layer 2, which has small pyramidal cells, layer 3 with bigger pyramidal cells, layer 4 with something called stellate cells, Doesn't really matter what they're called. It's just very clear that if you just do a kind of color by size, suddenly structure appears. But each of these cells has a three-dimensional structure. And here, for example, is a picture showing all the spiny motor neurons in this data set. So these are all the neurons that have spines. They're called excitatory neurons. And I'm going to just show you that in addition to getting each of these cells, because we've identified all the synapses, we can get all the synapses making contact with each of these cells. Here for a pyramidal neuron on the left, all these little dots are either excitatory synapses if they're blue dots, or inhibitory if they're yellow dots. We do that for excitatory neurons and interneurons. And not just for two cells, but for thousands and thousands of cells all at once. That's why connectomics is so powerful. So here are all the neurons in this data set. Again, layer 1 near the skull is here, and the white matter is down here. And I'm going to now show you again just the excitatory neurons. And I'm now going to spend the rest of the talk talking about just one of these cells, which is this cell right there. It's a little pyramidal cell in layer 3. And I'll just show its full arbor because you can barely make out its details in this picture here. So here's the cell body, and it receives input on its dendrites, which are spiny. These little things sticking off the dendrite are its dendritic spines. And here's its long axon that's going to leave this volume altogether. And here are all the synapses this neuron makes on other cells. This is the cells it talks to. And it establishes 695 synapses. Each of those yellow dots is one of them, to postsynaptic cells in the volume. And I'm going to show you those. So here is that cell that we just were looking at. And here are all the cells it talks to. And this is why understanding the brain is complicated. It talks to a lot of cells. 466 nearby neurons are getting connections from this one pyramidal cell. And that would be true for every cell. Every cell would have this complicated sort of motor unit, neural unit of target cells it talks to. But most of these connections are super weak. So 86 of them, 86% of those 695 synapses are target cells that only get one synapse from this cell. Each cell gets thousands of synapses, and this cell is providing one synapse to 86% of its partners. And it's providing two synapses to 9% of its, about tenfold less, to 9% of its target cells, 43 cells. And it's providing three synapses to even less. Only 13 cells are getting three connections. And only one cell receives four synapses from this cell. So the axonal output of this layer 3 pyramidal cell, it's very common for it to make super weak connections on target cells and rare to make stronger connections like 4 connections. But if you add up 86 plus 9.2 plus 3 plus 2, you don't get to 100%. There's still something missing. But let me just show you that those connections that I've just been talking about are based on the fact that the axon of this cell runs straight. It bumps into a dendritic spine of another cell, makes a synapse, and it just keeps running. There's no particular affinity of this axon for any cell. It just runs straight and it bumps into things and it makes synapses. So that's the vast majority, over 95% of its connections are like that. Well, what are the other connections? Well, there's one cell, 0.2%, that got 5 synapses. And there's 2 cells that got 7 synapses. That's 0.4% of the total of the targets. 2% of the targets, one cell got 10 synapses. That's a large number of synapses. And one target cell got 11 synapses. And one target cell got 17 synapses. And one target got 52 synapses, and one target got 53 synapses. So in addition to these very weak connections, there are rare, powerful ones that you would never really find except by virtue of connectomics, where you do this exhaustive search. And that turned out to be true for every cell we looked at. So let me just tell you very briefly about them. Here's the cell that gets 52 synapses, this purple cell from this one axon. It doesn't seem to have any special relation to it, but if you look when the axon passes the cell, instead of making one synapse, it makes, in this case, 6 synapses. And when it passes it at this site here, it makes another 4 synapses. Wherever they touch each other, this axon makes lots of synapses with the same target cell. In that case, like 10 synapses right there, and so on. So a very powerful connection, and it is interesting how different that is from the weak connections. And the inputs to this blue cell also have the same issue. You can ask, is there a strong input to this cell? Again, we get the connectivity of every input to this cell, and there are 2,230 of them. Most of them are very weak. 94% of the axons that innervate this only make 1 synapse on it. 4% of the set inputs make 2 synapses. And down here, you can see there's 1 axon that makes 9 and 1 that makes 11. So, and there's 1 of them. So this is now the input to the cell, not the output. So both the input to this cell has rare, super powerful inputs, and among its outputs, there are rare, super powerful outputs. So if this cell is typical, i.e. if all cells have 1 or a few powerful inputs and 1 or a few powerful outputs, then if a powerful input is activated, information may have a fast way to travel through the cerebral cortex without the need of doing things like recruitment or temporal or spatial summation. If you want to automatically move your foot off the gas pedal onto the brake pedal, you'd like to have a wiring diagram that doesn't require you to think about it, but just does it automatically. So, like in development, we have one connectome with 2 kinds of innervation, some strong, very intentional connections and weakly incidental connections that just happen where an axon passes a target cell. And might the brain have both these strong, fast, reflexive type pathways with super powerful connections and these slow, ponderous, thoughtful ones? And do we get to learning how to do something by transforming the latter, these weak connections, into the former, like synapse elimination at the neuromuscular junction? So I'm pretty much done now. I just tell you that this kind of work could go forward if we could do a whole mouse. And that is what the National Institutes of Health is trying to figure out. Is it going to be possible to do a whole mouse brain's connectome? That's 75 to 100 million neurons, 200 billion synapses. And it's about 500 bigger than the volume I just showed you. It's about 8 by 8 by 8 millimeters. And depending on how big the voxels are, it will probably be no more than 1 million terabytes or 1 exabyte, which is still 1,000 times bigger than what we've done so far. So that's maybe in the future. I'm happy to talk about it if any of you are interested. And I'll just end by saying I did most of this work, not at all. I just watched it go on, and I pushed people somewhat to try to get these results, but this is the hard work of a number of really amazing scientists, young scientists, many of whom are looking for jobs. Yaron Mirovich had a fantastic team of interns he worked with. Ryan Draft, who did the Brainbow work. Kate Kang, who did all the serial EM for the P0 mouse muscle. Alex Shapson-Coe and Daniel Berger oversaw the human data that I talked about. Richard Schleck did all the cutting and imaging. Juan Carlos Tapia did the early serial reconstructions. And then a really great team of Varen Jain at Google helped with a lot of this work, which could not have been done without their amazing skill sets. By the way, if you're interested in generating connectomic data sets yourself, we can help thanks to the fact we have a grant from the NIH, a U24, that allows me to use some of our excess capacity for projects you have. I hope it's not a big project, bigger than a cubic millimeter, but if it's a smaller project, please email me and I'd be happy to talk to you about that. So let me thank you all and shut off my sharing. No.
Jeff, thank you so much for a wonderful presentation, wonderful data, and please put me in this line of your U24 NIH grant for analyzing some not very big piece, but a little piece of tissue, as I said. Thank you so much.
My pleasure. My pleasure.
I know you have another appointment now.
I have to teach at 10:30. I don't, let's see, 10:04. So if there is a question or two, I'm happy to take it if you have time. If not, I understand, but I'm happy to take a few if there are questions.
There is already a question saying, how do you infer the firing order for rainbow data?
Yes, a really excellent question. And sorry, I didn't have time to go through this. We measured the conduction velocity of muscle fibers, how long it takes an action potential to get from one place to another. And we measured the conduction velocity of axons at these various ages. Then we built a model based on that, and then looked at various ages where the neuromuscular junctions were closer together, and all the data fit a model that was completely consistent with data like that. So we didn't actually measure the timing. We inferred the timing from the structure of who's connected to who. It's kind of interesting. There was no reason that the sharing should have been in a linear order, but it was in a linear order just as it was during synapse elimination at an earlier age. So we made an assumption that the same mechanism might be playing a role in both cases. But it is a fair criticism to say that it would be much better to do a direct measure of the firing patterns of each of these axons. If I could, I would. I just don't know how to do that. Connectomics is structure of dead material, but to do living recruitment orders of many axons is very hard, at least for me. So if somebody has an idea how to do that, I'm all ears. I'd love to collaborate with anyone who can figure out how to do that.
Is there any other question from the audience? Someone wants to write in the chat or open the microphone. There is a question from Kivit. How is synaptic plasticity taken into account in the number of connections each neuron receives?
A neuron makes and receives, or did you say makes?
Makes and receives.
Receives, yeah. So at birth, motor neurons make many more connections than they do later on, but the connections are weak. They're very small connections, and the muscle isn't very large. So in muscle, at least, the plasticity comes as a consequence of activity, not as the cause. The activity, the animal or the human is using the muscles, and probably true for brain circuits as well. You're using the circuits, and then connections are being left or disappear. Why an axon might, in the adult, make 52 synapses on one target cell and only one on a bunch of others is the question of whether, if you, in association cortex, make a strong association, somehow that target cell's activity is now in sync with the presynaptic activity, in part because once an axon makes 52 synapses, that axon in and of itself can drive the target cell to threshold. So it's sort of igniting. It's sort of, once it starts getting strong, it gets stronger and stronger, just as it does at the neuromuscular junction. Once an axon makes a certain number of synapses and gets above threshold, it makes even more synapses to have a huge safety factor at muscle. So I think there's a sort of positive feedback in the plasticity, but that's the consequence of activity, not the cause. And I think every axon probably among its targets has a few that are its favorite targets. And many of them in an animal like us that does a lot of learning are based on experience rather than on some genomic or molecular cue.
Any other questions? Jeff, returning a little bit to the rainbow data, do you think that the maternal, have you ever looked at the embryos, or do you think that the maternal interaction, connection with the embryo would be a role in it?
Yeah, our problem is that the expression of rainbow with the genes we're using gets progressively less impressive before birth. So we don't have a complete wiring diagram before birth with rainbow. But we certainly could do serial EM before birth because the muscle gets very small. But the hint we have, I didn't go into this, but I did mention that even at birth, the size of motor units range, even though they're all large, they range still tenfold from the biggest to the smallest. That range is exactly the same as in the adult, suggesting that even by birth, there already is some connectivity pruning, perhaps, if they were more similar earlier. And indeed, when we do the same kind of mapping of connections, we see the beginnings of a synfire chain, even at birth, that even though there's so many more connections, so it's a much noisier system, it still seems that muscle fibers tend to be innervated by a subset of axons that shift as you move along this firing chain, as if prenatal activity, the kicking of a baby, a fetus, human fetus, or a mammal fetus, even a chick embryo, is playing probably some role in getting this tuning up and running even before birth. And in most animals, they have to start walking shortly after birth. Humans are very fortunate. We have a year where somebody will carry us around. But for most animals, a lot of this has to be pretty much finished in the first couple of postnatal weeks, if not sooner.
And I was wondering, when you're presenting this very new idea about two neuromuscular junctions competing in the same fiber, have you ever looked in those? Because, as you said, the great majority of the muscles are the muscle fibers are innervated by only one neuromuscular junctions, but there are special muscles like the extraocular muscles that are polynervated throughout life. Are you looking at those? Because
yeah, the extraocular muscles are especially interesting because it's thought that they have a tonic end plate and a twitch end plate. And the tonic end plate may be in a part of the muscle that doesn't actually fire action potentials, but uses local contraction. In snake muscle, there are muscle fibers that do not fire action potentials. And they have, not surprisingly, multiple end plates along their length. That was the first thing I studied when I was an assistant professor. And not only that, not only do they have multiple end plates, but each end plate remains multiply innervated throughout life. That was my first independent paper. And that was interesting because this very same muscle also has twitch fibers that only have one neuromuscular junction and undergo synapse elimination. But the tonic fibers without postsynaptic activity, there's no way one neuromuscular junction knows another exists because there's no propagating activity. And there's also no synapse elimination at the neuromuscular junction because there's no postsynaptic activity that causes synapse elimination, I think. So I think that kind of multiple innervation is related to the lack of action potentials. What I was showing in this case were places where there are action potentials, but because the timing is so similar, they don't know each other is there. It's like two planets on the opposite side of the sun. They never see each other because they all, whenever they're active, their action potentials are bumping into each other. So they don't know that there's another neuromuscular junction on the same muscle fiber. So they don't compete.
And Greg here wants to ask you, are people doing connectomics on Drosophila or zebrafish or other Drosophila mutants?
Yes, so Drosophila at Genelia Farm, they have done the complete connectome of a mouse, of a Drosophila brain, and another brain, hemibrain, has been done. And that has transformed a lot of Drosophila neurobiology. So neurobiology in Drosophila is forever changed by virtue of the fact that in that animal, at least, there is a complete wiring diagram. It's not completely complete. They don't go all the way out to muscle and The sense organs are not connected, but it has transformed a lot. There's huge numbers of paper on Drosophila connectomes. In C. elegans, I was part of a group that published a paper in 2021 on 8 isogenic C. elegans. And that field also now has a lot of, and it's a longitudinal study of how that wiring diagram changes as C. elegans grow up through the various larval stages. And I think that field also is changed by virtue of having wiring diagrams now at many ages. So I think in these kinds of model organisms, it's profoundly impactful in the way people think about neural circuits. In mouse, it's not, because, you know, to do a whole mouse is going to be a big job, but there's a number of us who are pushing very hard to make the government figure out how to pay for something that will be insanely expensive and cannot be done in a laboratory. It has to be done institutionally at a much bigger scale.
And I can imagine how big is the bill that you already have, right?
Yeah, I think the thinking is this will cost, you know, in the range of $500 million with a B.
Insane. Any more questions? Anyone? Jeff, so it was not arranged by the two of us, but Santiago Ramon Cajal was today. I think it's clear the admiration that both of us have and how he's still inspiring us, right?
Yeah, fantastic genius who really set in motion a wonderful way of thinking about brains. Yeah, he's wonderful.
Thank you.
I guess I better go. I got to teach undergrads.
I'm positive you're going to give a fantastic class. Thank
All
you
right.
so much.
Thank you. It was a pleasure. Thank you for inviting me.
Pleasure for ours. Thanks a lot. See you.
Bye-bye.
Bye. So this is the end of our morning section, and we'll be back at 2 p.m. Brazilian time for this section about novel EM methods to study cellular and single particle audio structure. I'll see you later. Thank you.